The promise of liquid biopsies, detection of circulating biomarkers. Title: The promise of liquid biopsies, detection of circulating biomarkers. Introduction. The field of medical diagnostics has made significant strides in recent years, advancing the way we detect and monitor diseases in patients. One such innovation that holds immense promise is the liquid biopsy, a non-invasive technique that enables the detection of circulating biomarkers in bodily fluids such as blood or urine. Understanding liquid biopsies and circulating biomarkers. A liquid biopsy refers to the analysis of specific biomarkers present in bodily fluids, particularly blood. These biomarkers can be DNA, RNA, proteins, or other molecules released by cells or tumors into the bloodstream. Liquid biopsies are particularly promising for cancer detection and monitoring. Traditional biopsies, which involve removing tissue samples, can be invasive, expensive, and carry risks. In contrast, liquid biopsies offer a minimally invasive and real-time alternative. By capturing and analyzing circulating tumor DNA, these tests can provide accurate information about tumors that may be difficult to access physically. Advantages and applications of liquid biopsies. 1. Early detection of cancer. One of the most significant advantages of liquid biopsies is the potential for early cancer detection, even before symptoms manifest. As CNA can be detected in the bloodstream before tumors become clinically apparent, liquid biopsies offer a means of identifying cancer at an earlier stage, improving overall patient outcomes. 2. Monitoring disease progression. Liquid biopsies are invaluable tools for monitoring disease progression and treatment response. 3. Identification of minimal residual disease. After surgical removal or completion of treatment, liquid biopsies can be used to detect minimal residual disease, MRD. MRD refers to the presence of residual cancer cells that may not be evident through imaging or conventional methods. Liquid biopsies enable physicians to identify MRD, providing vital information for guiding further treatment decisions. 4. Personalized medicine. Liquid biopsies hold immense potential for personalized medicine, as they allow for the detection of specific genetic alterations and mutations in circulating biomarkers. By identifying these genetic alterations, physicians can tailor treatment strategies to the individual patient's molecular profile, improving treatment efficacy and reducing adverse effects. Challenges and future directions. While liquid biopsies have shown great promise, several challenges remain to be addressed. The sensitivity and specificity of liquid biopsy tests need to be optimized to ensure accurate and reliable results. Additionally, the cost-effectiveness of liquid biopsies needs to be assessed. As with any new technology, affordability is a crucial consideration for widespread adoption. Continued research and development efforts are required to reduce cost barriers and ensure accessibility to liquid biopsy tests for all patients. Looking ahead, the future of liquid biopsies is promising. As technology evolves, new biomarkers and detection methods may be identified, expanding the diagnostic capabilities of liquid biopsies. The development of novel techniques, such as next-generation sequencing and digital PCR, holds great potential for enhancing sensitivity and improving performance. Liquid biopsies, a non-invasive technique for detecting circulating biomarkers in bodily fluids, have emerged as a promising innovation in the field of medical diagnostics. One of the significant advantages of liquid biopsies is their potential for early cancer detection. By analyzing circulating tumor DNA in the bloodstream, liquid biopsies can identify cancer even before symptoms appear, leading to improved patient outcomes. Additionally, these tests are invaluable for monitoring disease progression and treatment response. Physicians can measure changes in circulating biomarker levels to assess the effectiveness of treatments, make informed decisions about therapy adjustments, and detect treatment-resistant mutations. Liquid biopsies are also essential for identifying minimal residual disease, MRD, which refers to the presence of residual cancer cells undetectable through imaging or conventional methods. After surgery or completion of treatment, liquid biopsies can detect MRD, providing crucial information for guiding further treatment decisions. Furthermore, liquid biopsies contribute to personalized medicine by identifying specific genetic alterations and mutations in circulating biomarkers. This molecular profiling allows physicians to tailor treatment strategies to individual patients, improving treatment efficacy and reducing adverse effects. However, challenges remain to be addressed for widespread adoption of liquid biopsies. These include optimizing the sensitivity and specificity of tests, standardizing methodologies, establishing quality control measures, and assessing cost-effectiveness. Continued research and development efforts are necessary to reduce the cost barriers and ensure accessibility for all patients. Looking ahead, the future of liquid biopsies is bright. Advances in technology may lead to the discovery of new biomarkers and detection methods, expanding the diagnostic capabilities of liquid biopsies. Techniques such as next-generation sequencing and digital PCR hold great promise for enhancing sensitivity and improving performance. The field of medical diagnostics has experienced significant advancements in recent years, particularly in the detection and monitoring of diseases. A promising innovation in this field is liquid biopsies, which are non-invasive techniques that allow for the detection of circulating biomarkers in bodily fluids like blood or urine. A liquid biopsy involves the analysis of specific biomarkers present in bodily fluids, with a focus on blood. These biomarkers can include DNA, RNA, proteins, or other molecules that are released by cells or tumors into the bloodstream. By analyzing these circulating biomarkers, liquid biopsies can provide valuable insights into an individual's health status. This allows for early detection of diseases, monitoring of disease progression, and the development of personalized treatment approaches. In the realm of cancer detection and monitoring, liquid biopsies hold particular promise. Traditional biopsies, which involve the removal of tissue samples, can be invasive, costly, and carry associated risks. In contrast, liquid biopsies offer a minimally invasive and real-time alternative. By capturing and analyzing circulating tumor DNA, these tests can provide accurate information about tumors that may be difficult to physically access. There are several advantages and applications of liquid biopsies. One key advantage is the potential for early detection of cancer, even before symptoms become apparent. CNA can be detected in the bloodstream before tumors become clinically visible, allowing for the identification of cancer at earlier stages and improving patient outcomes. Liquid biopsies are also invaluable tools for monitoring disease progression and treatment response. Moreover, liquid biopsies can identify minimal residual disease, MRD, which refers to the presence of residual cancer cells that may not be evident through imaging or conventional methods.